I am very happy to welcome our first speaker, and that is a user project for image data analysis. And we have Shiladitya Mitra as well as Michaela Blazikova, who actually took up the first image analysis project that came through Euro-BioImaging. So with that, please take it away. I should stop sharing my screen.
Thank you. Apart from the first slide, so I was actually trying to, if long story short, so I wanted to help with analysis. So we were trying to understand resilience and susceptibility based on the social defeat stress model, running them through two different paradigms, and then doing a whole brain CFOS mapping using Lightsheet. Where I was stuck was mostly the analysis part of it because I'm not an expert with analysis and clear map required. And the kind of analysis that was required was clear map after the light sheet images that were coming up. And this required a little bit of machine learning, a little bit of adjustments to refine the data and dig deep into it. This is where I required help. And at that point of time, I had a friend over there at EMBL, Rahul Surekha, and he gave me contact of Christian Tisha, who actually forwarded Astha's contact at that point of time. And Astha, thank you very much for coordinating and helping me in uh, come in contact with Michaela, who were very kind enough, and Michaela's group at Czech and Prague Node were very kind enough to pick up this project from there. And so I'll just give you a brief overview of what the project, what we intended and what the results we got and how the output looks like, while Michaela will go through final details of it. So just in a general brief overview, we were defeating the black 6 mice for 10 days, which induces depression-like phenotype. We were analyzing it through elevated plus maze and social avoidance to different behavioral paradigms to test for anxiety and social avoidance after being defeated. And based on these scores, we would differentiate them between resilient and susceptible. And then each of them would be taken for whole brain light sheet imaging with CFOS staining or a section by section immunohistochemistry, very old school style. We would compare this later on to come up with final targets that we would then go check for analysis of our odd red validation. So this is just a general slide over here. This is how we came up with a z-score from based on both these two behavioral paradigms, and the ones which are over here positive would reflect more towards resilient phenotype, where over here which are negative would correspond more to susceptible phenotype. We also employed deep lab cut and deep OF, which is our in-house um, package based on deep lab, which is a very popular deep learning based behavioral estimation tool. And based on that, we can see over here that before and after the social defeat paradigm, there was a significant difference between the groups. And when you differentiate between the control, resident, and susceptibility, you see that between the control and the susceptible groups, there is a significant difference, whereas a resilient lies somewhere between. And these are basically each of the different behavioral markers that we set while doing this deep analysis and how much component are coming into this analysis. But based on this, what we found that we could differentiate between resilient and susceptible and control very clearly, and we were ready to give our brains for further analysis. The first analysis that we did was basically an IDISCO-based whole brain clearing followed by a light sheet imaging. And here, this was helped by my ex-lab over there in Dansky. We used a 4x resolution and did the whole brain light sheet imaging, and then followed with the analysis to claim up. This is where I handed over to Michaela for the analysis. This is just a short video of how the whole brain CFOS staining looks like. But of course, it's much more refined when it comes down to section by section or optical sections. So yeah, so after I gave this data sets, one thing have to be taken into account over here is that the imaging was done from an hemisphere and not the whole brain. And we gave this data to Michaela after that, who ran this data through and gave us a plot and summary of the different regions of the brain and what are the CFOS. So CFOS is an immediate early gene, and we were checking for similar staining at different regions. So she gave me so she did the segmentation with the clear map and each of the different regions and how much CFOS was expressing in them. Then we did a short bioinformatics analysis, and we found that there were a couple of regions, and of course, more stringent criteria that were set over here. We found the ventral tegmental area of the ventral tegmental nucleus, and this region is already known for to be involved in this is dopaminergic region involved in stress and susceptibility, and these are basically dendrogram plots to show that how different modules were coming up. And this is the data from the whole brain clearing that Mihaela gave to us. We can see that the stress group comes up with different modules as compared to the control and the resilient groups. What was interesting that we did a section by section immunohistochemistry to validate our results. And when we combine the data that comes from the Lightsheet and the section by section immunohistochemistry and old school type and came up with the regions, we did find a lot of common regions which are put over here. The VTN was still showing up. We picked up an interesting region, which is the dorsal raphe, which is understudied when it comes to stress and resilience. And secant over here is not exactly significant, but there is a catch to that. And this was a technical limitation of the experiment on my part, because as I told that while we were doing light sheet imaging for the hemisphere, so this is where, because dorsal raphe over here, if you can see, it falls exactly at the midline. So when you are cutting the brain in half, so you are missing out sometimes on most of the information that's coming from dorsal raphe. And that's exactly the reason why we decided to stick to whole brain sectioning when it comes to section by section immunohistochemistry. We believed in that data, and based on both the data, then we decided to pick up dorsal raphe, which was coming up as close to significance in combined and significance in just a section by section immunohistochemistry data, and did a plot analysis to find that in the resilience, DR is not involved in any node, whereas the DR is involved in a very intense node of distress. So we decided to do a dread-based analysis for injecting an anterograde tracer into the anterograde virus into the dorsal raphe and retrograde into the anterior BLA, which we also picked up. The virus is supposed to recombine, and based on this dread or chemogenetic analysis, we found out, long story short, that we could rescue this stress phenotype that we are able to pick up that comes up during this 10 days social defeat stress. So long story short, in this whole project, we could identify multiple areas of the brain that are involved in stress and resilience and showing differential C-force activity that comes from the whole brain clearing. We checked it with section by section immunohistochemistry, found out common plots, and then did to a more functional analysis of a network. And of course, a lot of people to thanks from here, my entire team and lab. I was in Matthias's lab, so his team is fantastic to help. Everyone helped. And then for the enamel, we did also MRI analysis, which I didn't show over here. And for the Euro-BioImaging group, EMBL and Michaela particularly helped a lot with the data analysis. And for others, we have Monica Marcin for the acquiring of the data for the whole brain clearing, and Bianca for a part of the analysis for the section by section. Thank you very much. And maybe then any questions, I'd love to take it up, or maybe Michaela would want to go through this.
Thank you so much for your talk, Sheila. And that was fantastic insights into resilience mechanisms. So Michaela, please go ahead.
Thank you, Asta, and thank you, Sheila. So I will now just continue with what Sheila already told you. As he told you, we got the message from Asta that there's an Euro-BioImaging project on the image data. It came to also the Prague node, and I decided that it sounded really interesting for me to take over the project. So he had these Lightsheet images prepared, and he wanted to analyze them somehow. That was like the first information I got. We started discussing everything in September 2022, just for you to have a timeline how long this whole project took. Although the work itself, the image analysis was, of course, not that long, or did not take that much time. So there was 12 mouse brains with 2 channels, autofluorescence and CFOS. they were already stitched. So this was the data I obtained, or was ready to obtain. And they wanted to identify the C4 signal and then map the intensity number between the groups using clear map. And so after the discussion, what we needed to do first was checking the available hardware and software before taking over the project. So what we actually were quite lucky, because at that time we just started to build our data center. Here's the scheme, and what we're interested in, I was interested in, was this part that we have now, the fast storage and long-term storage, and on top of it, the virtual workstations accessible through the remote desktop connection. And that's actually what I was using in this project, a Linux machine that had access to this, to our storage. So this was definitely necessary, because according to what they say on the ClearMap website, actually, these are the minimal requirements. So that's what we needed. And then regarding the software, we needed a ClearMap that's written in Python 3, and actually it's available as Jupyter Notebook, so it's really easily accessible. At first, I tested on a small data set, actually on our data, just checking if I'm able really to get the software running and really obtain the results the user of Sheila was looking for. And after that, we agreed on taking over the project, and we agreed to test it on the project data, because these are quite sensitive data. So until this step, I was not able to see the real data that I was going to work on. Then in February 2023, so actually it's half a year, 6 months after we started discussing the project, the first project data were transferred, and I analyzed this data, and then we agreed on some other stuff. Regarding sending the data back and forth, we used the file sender from SESNet, so the resources we have in the Czech Republic, because these are quite large data, about 100 gigabytes each mouse brain. So then the final project, that was 12 mouse brains, so it was over a terabyte of data. Then I tested these project data using the ClearMap, and the aim was to annotate it to the Ellen Brain Atlas, as you see here. And the ClearMap is a pipeline that enables the automated analysis of brains. It enables to detect the cells, the C4 signal, and register it to the Ellen Brain Atlas automatically. So it's really able to do all these image processing steps, find the cells, register it, and then measure the shape and intensity of each individual cell. Now, the part that we needed to discuss with the user, with Sheila, was the segmentation first. And regarding the segmentation part, we're sending some results back and forth until he was satisfied with the segmentation part. We agreed that we'll use the segmentation available in the ClearMap, but there are some parameters that need to be set up. And then he also checked the annotation to the Ellen Brain Atlas. So the first image on the left is image from the Atlas that's actually transformed or registered onto the autofluorescence signal of the brain. So after we agreed that this is correct, so we agreed on also on the output, that the output will be the segmented cells, then annotation to the Atlas, and then finally, what they needed for the publication, or they need still, are the density maps of the brain areas and some mean density maps, and that was actually done already in MATLAB. So after testing the data, we really needed to agree on the final output. And after that, we were able to sign the contract. Regarding the contract, because this is really more a customer oriented project, so we needed to estimate the price, and we really needed to estimate how many hours the project will take in total. So after testing one of the brains, I came with the number 8 hours per one mouse brain, so that would be 96 hours in total. But this needs to actually include the preparation, some testing, then checking the output, creating the final results. So this was really some estimate, and then agree on the price per hour. After that, we finally transferred all of the data, all 12 mouse brains. I performed the segmentation, annotation, and the post-processing. We needed to check many things. Actually, one of these, like input, there was some issue with stitching, that some parts were not really that nicely stitched. We tested other softwares, but then we sticked with the big stitcher because the results was not really much better. Regarding the output, we needed to check the cell detection, the alignment, but that was checked by Sheila. So each of the 12 mouse brains needed to be checked by Sheila, actually. And after this monitoring and controlling phase, the final transfer of the results took place. Sheila checked all of the results, and now actually, and after now it's being paid, of course, and then now it's in preparation for the publication. And after that, we agreed that I will check all of the figures and methods, legends that will appear in the final publication. To sum up, it took from September 2022 until October 2023 to execute this project. The payment was for 96 hours in total for 12 mouse brains. In reality, I recounted down the hours so that we know how much this estimate coped with the reality, and it was really around 100 hours, something like 104 hours that I really spent altogether on the project, including the testing phase. So I think it was quite a good estimate. The issues that we needed to cope with really was the price per hour. This is a national level, and it was then estimated through the Czech bioimaging infrastructure. Then, of course, how many hours to count, how to include the testing phase. Regarding the data transfer, we were discussing first how to send the data, because there was quite a lot of sending back and forth, really through the fine-tuning the results through this monitoring phase. Bit issue with the meetings. Of course, there are many meetings online nowadays, but sometimes a bit complicated to meet and discuss everything. And regarding the publication, of course, there's an issue with becoming part of the project and becoming the co-author, or just being acknowledged in the paper. We agreed at the beginning that I will be just acknowledged in the paper. This brings less responsibilities and less duties. On the other hand, there's no space for testing some new approaches, like different segmentation methods, stuff like that. But definitely this needs to be agreed at the beginning of the project before taking over the project. Regarding the benefits, I think there's many benefits, not only for the user that needs help with some part of the project that he cannot really do himself, or would be just too difficult for him, but also for us, that these are really new image analysis approaches that we can then use internally in the future, and also we learn how to handle such external projects. And with this, I would like to thank our facility, our boss, Ondrej, our microscopists, Ivan, Mirka, Helena, and image analysts, Jan and Martin, and of course, Shiladitya Mitra, the user that came with the project. Thank you.